right, ladies and gentlemen, we're going to talk about mitosis today, a kind of cell division where two new cells are identical to the original cell. I have to talk fast because there's only about 10 minutes before I get cut off. So bear with me. If at any time you fall behind on note-taking, just pause the video and catch up on your notes. Okay, when I talk about mitosis, I often spit the letter T because it's cell division where you get two new cells. Meiosis is the other kind of cell division we're going to talk about, and that's where you get four new cells, and they are unique sex cells. These are identical cells to the original parent cell. All right, first of all, we need to talk about the cell cycle. The cell cycle is sort of like a life cycle. The cell is born and starts to grow during the G1 phase. During the S phase, its DNA gets copied, which means that it goes from having monovalent chromosomes to bivalent chromosomes. That's where those X's start to show up. And then the yellow sections of the diagram are where the cell is actually dividing. So the majority of the cell's life is spent in that blue phase, which is called interphase. There are, just as an FYI, there are some cells that no longer go through cell division, like the cells of the human nervous system. Once they've divided once, that's all they get to do. We have all kinds of feedback switches in our cells that control how many times the cell can go through cell division. So take a look at the diagram. At G1, the cell growth is assessed. At G2, your DNA replication is assessed. So are there mutations? Do they need to be repaired? And during the M phase, mitosis is assessed. How well is the cell dividing? If errors are found, the cell actually has the ability to try to correct those errors. Unfortunately, it says what might happen if the checkpoints fail and cell division continues when it shouldn't. That's what leads to cancer. So when we get mutations in the checkpoints that try to control our cell cycle. We'll talk about more about cancer on another day, on another time. All right, so mitosis is a series of events that occurs that are sort of part of a continuum. And we use the phrases interphase, prophase, metaphase, anaphase, and telophase to describe them. They're kind of hard to remember, so people often say the words IPMAT. And so my apologies to any mats in the audience. IPMAT, interphase, prophase, metaphase, anaphase, telophase. And those are pictures of what it would look like in a plant cell. Let's take a look at the different stages. The first stage is called interphase, and we already talked about that. That's the cell's normal growth period. So imagine a woman who can get pregnant. The majority of her life is spent not pregnant. So that would be her interphase stage. When she is pregnant, however, is what starts with cell division in my analogy. So prophase would be the first stage of pregnancy, if we were going to say it that way. Anyway, interphase is when the DNA is in the chromatin form, so it hasn't wrapped up to form those chromosomes yet. But the DNA does have to copy itself during interphase. So during prophase, that's when the DNA copies itself, the nucleus disappears, and you start to see those X-shaped chromosomes. They have things that are called centrioles, those black and white organelles, move to the poles of the cell. And then they start to grow those finger-like projections that are called spindle fibers. The purpose of the spindle fibers is to shove those chromosomes into the middle of the cell. When we look at prophase under the microscope, it has a tendency to look polka dotty, and that'll start to make sense to you. I'm using a little alliteration. Prophase is polka dotty. The next stage, called metaphase is in the middle, metaphase middle. So that's how you're going to remember the metaphase stage. So the chromosomes have been pushed into the middle by the spindle fibers, and so they're lined up along what's called the equator, and we remember that by metaphase middle. The next stage, those spindle fibers pull those chromosomes apart so that they're kind of facing each other. Remember that the two sides of any chromosome, so like an X chromosome, for example, the two sides of it are identical copies. So when you pull half of it to the left and half of it to the right, you're pulling identical copies to either side. So now when this cell does divide, identical DNA goes into the left cell and identical DNA goes into the right cell. So this stage is called anaphase, and we say anaphase apart as the chromosomes are being pulled apart. The last stage of mitosis is called telophase, and that's where you get two new nuclei. And then finally, there's another stage that's called cytokinesis, and this is when the cell membrane actually pinches in. We joke that this picture looks a little bit like a baby's butt, but that's an animal cell that's pinching in. In plant cells, cytokinesis has to look a little bit different because plant cells have that hard cell wall. They can't pinch in. So instead, they form something that's called a cell plate. And that's kind of how we define the end of cell division, is when that cell plate forms between the two new nuclei. All right, so interesting questions. Why do cells die and why do we age? And it all has to do with mitosis. A vocabulary word that's important that you understand is something called apoptosis, which is programmed cell death. Cells are only allowed to divide approximately 50 times, and 50 times we call the Hayflick limit after the scientist who discovered it. So that's normal cells. They can only divide 50 times. However, we have found that cancer cells can continue to divide. There's a very famous case of a woman named Henrietta Lacks who had cancer. Her cells were removed from her, I believe, in the 1950s, and her cells are still dividing in science labs today because they were cancer cells, so they can continue to divide. So the question is, do cancer cells hold some kind of a secret to increased lifespan, even though they're cancer cells? And there's a possibility that they do. Part of aging is when our cells stop dividing and they don't replace themselves. So could we somehow learn something from cancer cells? And the answer is probably yes. All right, so let's take a look at these diagrams, see if you can name the phase. I'll give you just a second to take a look at them. Probably the easiest of the stages to name is the one in the upper left-hand corner. You've got two cells there, so that would be telophase. The next easiest is maybe the second from the left on the bottom row. Those chromosomes are being pulled apart. Alliteration here, so apart is anaphase. Then the one to the left of that is metaphase, metaphase middle. And then the last two on the right, the first one is interphase, and the one that looks polka dotty on the far right, that's prophase, so polka dotty prophase. I really encourage you to take a look on your notes. These are live links. Try the Onion Mitosis Labs 1 and 2, where you can practice looking at the different stages of mitosis and see if you can get them right. All right, ladies and gentlemen, that is it. Next set of notes is on cancer.